I'm going to go over the mitosis study guide with you now. You'll just follow along and check your answers. By what process is DNA copied in order to make new cells? Well, before a cell can start dividing, it has to take that chromatin that's kind of jumbled up like a bunch of spaghetti in the nucleus, and it has to copy the DNA. There is stretched out like spaghetti because that makes it easier to unzip it, open it up, and make copies of that DNA. And that process of copying the DNA is called DNA replication. So DNA replication would be the answer. Okay. Number 2 says, in which phase of mitosis do the chromosomes divide and move to the opposite poles of the cell? And that is anaphase, and we remember that ana starts with A and away starts with A. So at anaphase, those chromosomes are going to move away from each other. So anaphase. And we're going to remember ana, away. Chromosomes are moving away from each other during anaphase. Number 3, if a human cell contains 48 chromosomes, divides by mitosis, and the daughter cells then go through mitosis as well, the cells that result will have how many chromosomes? Well, the process of mitosis makes identical cells. So all of the cells would have 48 chromosomes. They would be identical to that original parent cell. So the daughter cells, after it splits, the daughter cells would have 48 each, and then after they split, those 4 cells would have 48 each. So they are identical. Mitosis results in identical cells with identical numbers of chromosomes. So 48, and we put identical, so we remember. Identical means they're just alike. Okay, so what biological process accounts for our skin healing after a scrape or cut? Well, that would be mitosis. Mitosis is making new cells to fill in that cut, that gap in the skin cells. Those skin cells undergo mitosis, making new cells, and then that heals the cut, fills in that space. Explain the main function of the spindle fibers. Well, way back when we first started talking about cell organelles, we talked about centrioles, and we just said at that point that centrioles are important for cell division. Right here is where they become important. The centrioles are going to spin out those spindle fibers, kind of like fishing lines, and those spindle fibers are going to attach to the chromosome when they're lined up during metaphase. They're lined up down the middle. Those spindle fibers are going to attach. They're lined up in pairs, so you have spindle fibers attaching on each side. Centrioles have moved to opposite sides, so spindle fibers are coming in from each side to attach to those chromosomes. And then those spindle fibers, much like a fishing rod, is going to kind of start shortening like you're reeling in your fishing line. And it's going to pull those chromosomes apart. So the function of the spindle fibers is to pull those chromosomes apart during anaphase. Remember, anaphase, they're moving away. So it's going to pull the chromosomes apart. The spindle fibers will pull the chromosomes apart during anaphase. Spindle fibers will pull the chromosomes apart during anaphase. Number 6, at the end of mitosis, the offspring have equal, half, or more genes in the parent cell. Mitosis is making identical cells, just like that parent cell. So if the parent cell started out with 48, the offspring are going to also have 48. So they're going to have the same number, equal number of chromosomes. Number 7. What cell organelle is important in cell division? Well, that's the centrioles we were just talking about that are making the spindle fibers, and that's the only thing we told you about centrioles back when we were talking about organelles. We kept saying, Well, that's the centrioles, and they're going to be important in cell division. We'll talk more about that later. Well, here we are. It's later. So we're talking about those centrioles. And those centrioles make those spindle fibers that are going to attach to the chromosomes when they're lined up down the middle at metaphase. And then during anaphase, as they're moving away, it is those spindle fibers that came out of those centrioles. They're going to be pulling them apart. Okay, number 8. Which phase of mitosis can be described by chromosomes appearing with sister chromatids and the nuclear membrane breaking down? That's prophase. That's early on. That's after the chromosomes have replicated, and now they start bunching up into little X's. Like the little X's that we can see. Well, X's is actually the pairs of replicated chromosomes, so like little X's. That's the first time that we can begin seeing those chromosomes. So homologous chromosomes are going to pair up in just a few minutes, but right now they're coming where we can see them. The phase of mitosis can be described by chromosomes appearing with their sister chromatids and the nuclear membrane is breaking down. Okay, so that's all happening very early. That nuclear membrane breaks down during prophase. So this is prophase. Remember PMAT, prophase, metaphase, anaphase, telophase. Prophase has to happen first. We have to get everybody out of that nucleus so the rest of it can happen. So prophase is where that nuclear membrane is breaking down, and it's when we can very first see those chromosomes begin to bunch up. So that's prophase. Okay, number 9, sister chromatids are attached to each other at what structure? They're attached at the centromeres. Centromeres are important because that's what we count when we're determining how many chromosomes a cell has. So when the chromosomes have replicated and they look like little X's, usually the original chromosome will be one side of the X and the newly replicated chromosomes can be the other side of that X when we see the chromosomes looking like little X's. But we only count that as one chromosome because they're joined together in a centromere. Okay. So we only count those as one at that point. And that's where they're going to separate. So if you have 48 chromosomes, those 46 chromosomes are going to have to replicate to be able to make an identical offspring there, identical sister cells. So they have to copy themselves, but we don't say, oh, now all of a sudden we have 92 chromosomes. Now we still count those as 46 chromosomes because they're still joined together at that centromere. Centromeres are what scientists, doctors, people count to know how many chromosomes are there. So centromeres, that's that little thing that's holding the middle of the X together there. And one's the sister chromatid, the other side's their sister chromatids where they've copied. They're identical there. So that's where they're held together. So cytokinesis. Cytokinesis is talking about the cytoplasm. So cytokinesis is the splitting of the cytoplasm as the cell divides. Splitting of the cytoplasm, cytokinesis. Number 11. During which phase of the cell cycle is protein production high and the cell is rapidly growing? Okay, so which phase of the cell cycle is protein production high? Okay, we say cell cycle because this is not actually part of the PMAT. It's not actually part of mitosis, but it is part of the cell cycle. Okay. So we have interphase that actually happens before prophase starts. So interphase is the time the cell's kind of resting, copying its DNA, making new ER, making new Golgi, all those things it's going to need, all those new organelles that we say makes eukaryotes take longer to replicate or longer to divide is because they have to make all those organelles. So that's what it's doing during interphase. It's growing. It has to have a lot of protein to be able to make all these cell structures. So interphase is actually happening before we get to prophase. Metaphase, Anaphase, and Telophase. Before we go into that PMAT, we have interphase. Talk about interstates. Inter kind of means between. So I think about interphase happening between divisions. That's when it's growing and making all those new parts, replicating its DNA, doing interphase. Number 12, what are somatic cells? Well, somatic just means body cells. And body cells are the ones that are doing all this mitosis. When we talk later about the other cell, about meiosis, that's not going to be the body cells. That's going to be gametes. But somatic cells, that's our body cells, and body cells do mitosis. Somatic cells are body cells. Just anything in your body that is not an egg or a sperm is a somatic cell. Number 13, during which process is unregulated cell division occurring? If it's unregulated cell division, cells dividing out of control, they tend to make masses of cells, or they push, and like if it's happening in the bone marrow, they're going to push out the good cells that need to be there. So that is called cancer. Cancer is unregulated cell division. Something's happened that the cell cycle doesn't cut off where it should. Cells are dividing out of control. That would be cancer. Number 14, this is the structure that forms between dividing plant nuclei during cytokinesis. Okay. Plant cells have the cell wall, so it's not easy for it to just pinch in two like the animal cells do. So when a plant cell is undergoing cytokinesis, it makes what is called a cell plate. And then that cell plate will become part of the cell wall for those sister cells. So cell plate. Number 15. Be able to recognize the pictures of the different phases of the cell cycle. We have done several different things, several different activities, and taken notes on those this week. So go back and look at the pictures that you have. Make sure you know what prophase looks like, what metaphase looks like, down that lined up down the middle. Anaphase, they start pulling away from each other. The spindle fibers are attached, it's pulling those chromosomes away from each other. Telophase, that you have a bunch of chromosomes at one end, you have a bunch of chromosomes at the other, and they're pulled all the way away, and it starts forming a new cell membrane around those. Telophase, and then it starts pinching in to, it goes into cytokinesis, which again is not considered part of mitosis. But it's the very end phase where the cytoplasm pinches into, then you have two distinct cells. And then it goes back into interphase, and that's when you've got all the spaghetti looking stuff in the nucleus and it's replicating that DNA. It's got to stretch that DNA back out so it can get in there and make copies. And that's where protein manufacturing is going to be high. That's where you're going to be making new endoplasmic reticulums and new Golgis and new lysosomes and all those things. It's got to copy. Okay. So all that's happening during interphase. And then when those chromosomes, those newly replicated chromosomes, start bunching back up and they look like little X's that we see in pictures sometimes. Well, that's the two sister chromatids joined together there, and they're joined at the centromeres, so we still count them as one chromosome each. Okay. And then they line up down the middle. That would be metaphase. Meta means middle. Meta means middle. So they're lined up down the middle. And then they start, centrioles attach spindle fibers. They start pulling away from each other again. Ana means away. Okay. And then we have telo. So at telophase, we have two separate areas of chromosomes beginning to make two new nuclei, about to divide into two new cells. And then it's back to telophase, 2T. Okay. So PMAT, prophase, meta means middle, ana means away, telophase means two. We almost have two new cells there. And then it goes into cytokinesis and interphase again. And that's the cycle. So make sure you know what those look like.